Let's thank Olivier again. Streichan from UCSB, and he's going to tell us about mapping forces, flows, and kinetics in the Drosophila embryos.
I'm taking your computer.
Yes.
Sorry.
Thanks for the intro and thanks for the invitation. Putting together this nice workshop. So, in the introduction, you were talking about the man who makes this field, and Darcy-Thompson. And we are, of course, also very inspired by Thompson's work on growth and form. And Thompson has been talking a lot about growth. In the end, he has a very interesting chapter where he talks about what he calls theory of transformations, which he put forth as a kind of explanation for how evolution of the form of organs may have come about, right? So he's comparing sort of fish to fish and all this, and sort of seeks for a mathematical framework in which he could try and understand this change across biology. And of course, he was very ahead of his time, and now we have many more methods at our disposal to come back to this old question. And what you find if you're looking at developing embryos, as we've seen in many of the examples today, is that there's actually quite a lot of reshaping going on. So this framework of theory of transformations may actually be more about development than perhaps about evolution. And so with modern techniques, we can now see entire embryos and organs. So this is, for example, a fruit fly embryo, and this is an organ inside this fruit fly embryo, and you can see how they are undergoing like dramatic changes in how they are configured. And this is what we would like to, by a mix of quantitative data analysis and physical modeling, together with experiments, would like to understand better. Some of the tools or some of the insights that haven't been there when Darcy-Thompson was around, but for example, how actually these embryos are being patterned, right? So how these biochemical processes that are running inside an embryo are organized. And so there has been actually this genetic revolution in the 70s, and some of the founding fathers in this field is who basically deciphered how in the line embryo we were establishing the fate that eventually sort of leads to this elaborate body plan that we're seeing. And so what you can see here at the very top is the fruit-like egg. And so this egg is basically on the anterior to the left, posterior is to the right, dorsal, which is going to be the back is to the top, and ventral is going to be the bottom, and that's going to be the egg. And so you may think maybe in the shape you already sort of specify some asymmetries, but that's not true. It turns out that the mother actually deposits information in a symmetry-broken way. So for example, here you can see the maternal morphogen decoy, which is deposited as RNA in the future head region, and then it's been eventually translated into a protein. This is establishing then this gradient pattern that you can see here. And this gene decoy is very important for establishing the fates of the head in a fly. So if you're having a mutation in this decoy gene, you're essentially going to make a fly that doesn't have a head. So it's not viable to survive. So together with a bunch of other morphogen gradients, decoy triggers zygotic information in this embryo. So now it's the genome of the embryo that's taking over, and this gets partitioned into sort of finer and finer levels of decay. So for example, here you can see hunchback and cripple that are basically two of these gap genes that are sort of expressing a larger domain, but it's not quite the scale of the decoy gradient which sort of covers the whole embryo. And these are called gap genes because if you mutate those away, you have a gap in the body plan. Entire parts of segments are going to be missing if you remove those parts. And this goes further down to genes that are pair rule genes that are basically called paired, E or RUND, and there are many other examples that are basically going to get rid of every other segment in the body if you have mutated them. And then finally, you're going to arrive at sort of these genes called segment polarity genes that are going to be found at the edge of a segment, for example, and that are down to a single cell precise. So there's basically an understanding that this anterior-posterior axis through this process gets patterned in a very precise way, so that down to the single cell you know where you are. And in a very similar way, you can think about the dorso-ventral axis of these embryos. So this is now a section to this embryo, and it looks like a cylinder on the inside of this kind of hole. This is going to be the back, this is going to be the belly, and if you're now looking for different genes, you will find them also being expressed in stripes going from the dorsal end to the ventral end. And so if you're thinking about this in terms of sort of mathematical languages, you may think of this as a coordinate system that is sort of patterning the surface of this embryo, right? And so these genes are playing a really important role in the process of how the shape of an embryo is going to be like. And this was very clear from looking at mutants of so-called Hox genes. These are master regulatory genes, where you can take a single gene, you mutate it, and you have a rather drastic effect in the form of the animal that you're finding. So this is head-on, a fruit fly adult now, and you can see here two antennae kind of sprouting out of sort of where you would have your nose as humans. And when you take this gene antenna P and you mutate it, what you see coming out of this is, instead of an antenna, now a perfectly functional pair of legs. So apparently by mutating a single gene, you can lead to drastic difference in the transformations that you're seeing in such embryos. And this is not just one random example. There are other examples. So for example, there's this ultrabithorax genes, which take the hull tears in these flies, which are tiny little balancing organs next to this beautiful large wing. And if you're a mutant for ultrabithorax, what you're going to get is essentially a duplicated structure of the notum, and then you have 4 of these wings rather than 2. So it's clear that these genes are playing a role in the process of these transformations, how you're sort of building a final shape. But so what we need to do now is basically understand how we get to these shapes and how these genes are eventually then going to instruct these shapes, right? And so, yes?
It might be unrelated, but for the flies, if you make 4 rings, can they learn to control them?
Yeah, I think they can actually fly, but I think they're much worse in flying than their friends, right? So their in-house friends, I mean, okay, the ones we have in the lab are anyways very bad at flying, yeah? And then those ones with those 4 wings are kind of even worse than the ones that we would normally see, I guess. Okay, so these are 2 views looking onto an embryo, and you can see now, sort of with modern microscopy, that we can actually observe all the cells in such an embryo at reasonably high spatial temporal resolution. Here to the left, you can also see that we are able to visualize components of this network of transcription factors that are acting in it. So this is even skip. This was one of these pair rule genes. So you will see 7 of these stripes occurring here, and they're initially rather shallow. They build up, and then you can see as these cells are moving along the surface, these genes are kind of following these deformations that you can see on the surface of that embryo. And when you're looking at the scale of the entire embryo, this pattern might not look so complicated. So when you're looking now at a microscopic level, however, you can see that there are also a very small set of behaviors that those cells have at their disposal in order to fuel this kind of thing. So for example, what cells can do is they can change their shape, they could grow or shrink, and this is what happens here in this midline as you're looking at the middle of this picture here. This is a ventral view onto the embryo. You can see this black line. Their cells are changing their shape, and then the mesoderm sort of folds into the tissue. Then they can interpolate. This is a movement that happens predominantly in the germband, and so they can change their neighbors. And then finally, they would have division or death at their disposal in order to sculpt this animal. And so, what we would like to understand is how you can bridge the gap between this microscopic level of understanding that you have about what the cells can do and the macroscopic outcome, and that's being sort of the shape of the whole animal, and how that would be instructed by these gene expression patterns. And so the way that I'm going to sort of distribute this today is into 2 pieces. So in the beginning, I'll talk a little bit about technology development that we have done so that we can take on this whole organ perspective, and that we can sort of take data like the ones that I've shown you. And then in the second part, I want to move towards the scientific question, what we would actually need to measure in order to predict these flows. So let's get started with light sheet microscopy. So I'm sure that most of you have already heard about it, and that this technique is kind of reasonably old in the core of its idea. So this goes back to Zsigmondy and Siegentopf, who've been building this thing called the ultra microscope, where their core realization was basically that if you're creating a thin sheet of light, and you're looking onto that sheet of light, that you have now superior background to foreground, and that you can have now detection of very low events, like scattering of a photon on some gold particle or something. And so they basically, in this way, measured the diffusion of gold particles. And so in the modern days, we're using this same idea. We're basically having a bit more fancy optics. So we have a scanning mirror, some scan lens, a tube lens, and some objectives so that we can generate Gaussian beams. And we can move these Gaussian beams rather rapidly so that we are going to generate a light sheet. And then we have an objective that is going to look onto this light sheet. In that light sheet, we can then basically park a sample. And if you're thinking now about the fruit fly embryo, you basically illuminate this from one side, and this beam moves through the embryo, and you can then take a picture of this and see a two-dimensional snapshot. Now, like every biological sample, there's going to be some amount of scattering present, and that means that you're going to get a systematic degradation of your beam as you're passing through the embryo. You'll have a very nice excitation on this side. On the other side, you'll have the beam fanned out, and so the excitation will not be quite as nice. If you're doing the same thing from looking orthogonally onto this sheet, you will suffer from the same scattering, so that the picture is going to degrade in a way, and you'll see 1/4 of this embryo reasonably nicely. So this is what you can see here in a cross-section through this fruit fly embryo. You can see all these nuclei sitting there. And so what we then did is basically put together 2 of these microscopes, right? Rather than rotating the samples 4 times around, what we're doing is we're having now 2 opposing excitation arms and 2 opposing detection arms. And together they define 4 branches. So there's an excitation and a detection arm, which basically would record this image here. And then there's another excitation and detection arm, which would record that image there, and so on and so forth. So then you have 4 of these different images. They are taken all in a different frame. So what you need to do then is solve computational problems so that you can map them all into the same frame, and then you can synthesize them together to get a single high-quality image of the whole embryo.
Is there a problem in resolution, or is there a difference, maybe I should ask, the resolution in the plane compared to out of the plane?
That's the next thing. So basically, Maha is asking, sort of, is there some anisotropic point spread function that you're having, right? And so this is, in a sense, a wide field microscope. So yes, it has an anisotropic point spread function, and this point spread function is basically going to be long in the direction of detection because of that aperture that you're having in the detection objective. And this has consequences for, when you're recording data. So, you look at your embryo and you can see how on this embryo there may be this thin structure, let's say membrane, right? This membrane basically grows in the normal direction towards the surface of this embryo. And as you're sort of going around the globe, you'll see how the membrane has now an orthogonal direction to the point spread function, whereas here it's parallel. You can look at this sort of an unwrapped version of this. What you see, as long as the point spread function is parallel to the membrane, you get a reasonably nice picture, right? But when the point spread function is orthogonally oriented to the membrane, it basically smears out everything, and that's where you get this kind of blurry picture. The way around this is now you take your sample and you rotate it by 90 degrees. This is effectively like rotating the point spread function around with respect to the animal. You're getting then a nice image in the regions that you had blurred before, and if you're doing that fast enough, you can again synthesize a very high quality image, and this time you're working towards an isotropic point spread function. So now you're going to take data with such a microscope. So these are taking whole embryos, sort of 0.5 millimeter long, and you're taking them at sort of 1/4 micron resolution. So an individual image is already multiple gigabytes in size, and if you're now doing this for the whole developmental time course of an animal, you're ending up very quickly producing multiple gigabytes of data. So you're facing a new kind of problem here. So, this is like the workhorse microscope that people have in their imaging facility. It's able to produce about 100 gigabytes per day. It's this confocal microscope that has been mentioned a couple of times. And here you can see a light sheet microscope getting assembled. And what this is producing is maybe around 10 terabytes a day. So if you do the math, you will see that within 1 week of imaging on this microscope, you've produced roughly the same amount of data as you could produce on a confocal in a year. And so you need to really revamp your kind of strategies, how you're going to work with this data. So you need not only have a fast network for infrastructure, but you also need to think about how you're going to store all this data, right? What are you going to do with all this stuff? And so one of the solutions that you could come up with is basically compressing your data, right? So that would be one approach. The advantage of this is that it would make the data size much smaller, which has the advantage that you can share it faster with your friends. But it has the downside that it comes, at increased access times, and then there is this very difficult discussion about do you do lossy compression or do you do lossless compression? So lossless compression schemes generally don't perform quite as strongly, so they only reduce by a factor of 4 or 5 or so. If you do lossy, you can get very strong compression, but depending on the problem that you want to solve, you're losing structure, you're losing information, and you can't quite approach it. And so then the last problem is still not solved, and that is that biological data is actually not organized in a way how we structure microscopy data. So when you're looking at this embryo here, you can see these nuclei sitting on the surface of this embryo, and they're now color-coded for depth. And you can see that this is a thin shell that is basically rotating. It's looking a lot like the surface of a rugby ball. And so this is an entangled layer, right? And analyzing such entangled layers is something that one can consider. So what we have then done is we said, okay, if we have such a situation, which is pretty general, it happens very often in biology that you have layers, then you can actually define a surface of interest. And you could say you just want to have the information that is on that surface of interest. So normally you have this cubic region of interest. There's corners in there that you're not really interested in, right? The yolk, which is sort of the inside of this embryo, may play an exciting role, but if I'm interested in studying the distribution of a molecule on this gray surface, then I would only want to actually know what happens on that gray surface. So I can represent these surfaces of interest in the form of a triangulation, and that's going to pay off because that's very general. You can use this for many different types of shapes. And then you can define a coordinate system on this triangulation, and you have a relationship between the plane, right? And you can do this in many different ways on the surface of an embryo, and you can then assemble what you would call an atlas for Earth, for example. And so we call this tissue cartography, and publish this as an image surface analysis environment, which is a freely available tool for everyone to use. So basically what you do here is you take your sample, this is my fruit fly embryo, and you partition it into multiple different domains. And you have an anterior domain, a posterior domain, and then you can take whole cylinders. The cylinders you get, for example, by making a cut on this green line there. And then you have coordinate systems defined on all of these domains. You can make the pullback of the fluorescence data into the coordinate plane. And what you're then seeing is basically a panoramic overview that shows you this whole embryo in one look rather than having to rotate it around. And you can make those domains overlap so that you have transition zones, and in that way you can then basically follow those objects as they're navigating across the globe. And this kind of global overview also simplifies data analysis dramatically for you, because there are tools available for segmentation and all that stuff.
Is the goal here just visualization, or you actually want to do quantification of?
We want to do quantification, and so in 2 dimensions, segmentation is orders of magnitude easier than in 3 dimensions. So identifying objects in 3 dimensions is a much, much harder problem, I would say, than finding them in 2D. There has been a lot of history working with 2-dimensional images. Most of the images that you see, sort of that Google is working with, are also 2-dimensional images, so there's a lot of technology around there. And in this way you can leverage, you can sort of tap into this technology. 3D is still very much in development. So, okay, yeah?
Yeah. How does the performance of this vary with the density of cells that are present on?
I think that's a question about resolution, right?
Sure.
So if the resolution is crappy, then you have a hard time segmenting the data. Yeah, that's absolutely clear. Yeah,
Right. No, I meant more like, if you have a lot of nuclei, then you actually have a lot of seed points for your mesh. I don't know how you're doing mesh,
think about
but then when you put it in
potentials.
2D, it's actually hard to separate segment dense nuclei, right? So
Yeah, so yes, so you can actually take, turns out, sort of multiple layers on top of one another, and then you can, this is like a matroshka, like these Russian dolls, right? And you can, or we call these onion layers, and put them down into some unfolded form, and then you can run filters over this so that you can find your objects much, much easier, right? So that, if you're interested in that, I'm happy to tell you some more details. Okay, so if you do measurements in these distorted maps, that's of course not going to work out nicely. So you'd see that most of your biophysical measurements, like measuring the area of something or the track, are depending on position, and that's because the distortion is non-uniform on these surfaces. But if you're just doing differential geometry and using metric tensor, you can actually correct for these, and then you can get very good agreement between the standard biophysics measurements in 2D as you would do them just directly in 3 dimensions. And so you may say, okay, fine, so I have this fruit fly embryo, the shape of this embryo is relatively simple, what if we have some more complicated form, right? And so this is a beating zebrafish heart, and you can see two views, so this is front view, this is side view, and these lines are basically showing you where the cross-section is taken from, and this is data from Jan Huskin's lab. So you can see the myocardium and the endocardium labeled in different colors, and you can see the zebrafish heart beating quite frantically, so there are two additional layers of complication. One here is in the shape is a bit harder, but then also the shape is dynamic. You know? So I said that it would be good to use triangulations, and indeed triangulations are able to recapitulate these shape changes that you can see as the heart is beating. You can then go the same way as you're doing in this fruit fly embryo, chop up the heart into multiple different domains. You can define your coordinate systems on each of these domains, and then you have a relationship into the plane. Now you've basically been diving into the tissue frame, and following objects in the tissue frame is now much, much easier. So you can track these cells, and then again, sort of your 2D analysis tool is better than, you know, or easier than doing it in 3 dimensions where you need to follow these objects quite complicated paths.
Don't you have to undo this distortion? I mean, this,
Right,
not
that's what
just
we
a
do in the
matter of
metric tensor,
scale.
right? So that's, but with the metric tensor we can do measurements. We can then account for how local areas and local angles have changed.
But what about the shape? I mean, it's a projective transformation. So it's actually not just, it's not just 1 or 2 scales. The whole shape gets distorted.
So what you have is, you have a relationship between the coordinates in your flat space and your curved space. And you can calculate any object, any kind of physical measurement with an object called the metric tensor that basically encodes the relation between this 2D and 3D. You know? And then you can make angle measurements, you can make fluorescence measurements, and this is all sort of encoded in how you spread out everything locally or how you distort angles locally. That's pretty broad. Okay, so to summarize this, what we basically now have is a couple of tools at our disposal that allow us to see whole embryos, very high spatial temporal detail, and then, you know, in order to handle this data deluge, we use this tissue cartography that dives into the rest frame of the tissue, which makes data handling much easier. These data are now 1,000-fold compressed, basically, and the quantitative analysis on these complex shapes becomes now also much more feasible in 3 dimensions.
Don't you need the curvature tensor as well? If you flatten, if you have the metric, you don't know the mean curvature.
If I want to do simulations or something like that, right? If I want to do the inverse, then I will need curvature. But for measuring lengths and stuff, we do that with the metric tensor. Okay, so let's get into the second part, and that's basically, can we actually do something with all this machinery that we've now developed? You know? And so this is a video showing a fruit fly embryo taken on this light sheet microscope, where you basically have some 0.5 millimeter length. This is the future head, this is the future tail, this is the back, this is the belly. This is just the other side, and each of these little dots are showing sort of the nuclei within these cells. And you can see how basically there is a very dynamic process on the road. So initially you have these 6,000 cells that are not structured in any crazy way, and now here you can see how actually structure has emerged, right? You can see these individual segments, and if you carefully counted them, you will find that there are 14 of them. And you can see that there, maybe you can't see in this particular video, but you can see that there are also inner organs now being assembled, right? So there's basically a gut there, there's a brain, there are muscles, there's a heart, and there is a trachea that's making the breathing. And so, you know, we would like to understand how, you know, these movements that you can observe on the surface of this embryo are being controlled, and so we would like to know where the forces that are controlling these movements are actually emerging from. And so this is work, of course, that has been sort of studied by many people before, and again, somebody who's done a lot of work in this direction is actually Eric Wieschaus, who got interested in this question of how you control the forces that deform the embryo. And one of the first and most striking events that you can see in this gastrulation stage embryo is that it basically will form this ventral furrow. So this is going to be in the ventral region, and there's going to be genes expressed in this ventral region. The two particular ones that we're interested in here are twist and snail. They are demarcating basically the mesoderm, and they're going to pick out this domain that is undergoing a major shape change. So if you're looking at this embryo in cross-section, you can see this domain here where twist and snell would be expressed. And the cells in cross-section would look like rectangles. And the apical surface in this picture now points down. And if you're looking slightly later, what you will observe is that the apical surface of these cells is going to change. It's going to become smaller. And the basal surface is going to expand. That's the other end of the cell. And this is going to continue until this sheet has basically completely folded to the inside. And this is showing some micrographs of this, where you basically see the expression domain of this gene twist. You can then see here the apical surface of these cells basically changing, and then this tissue basically has fully folded in. This is like a piece of paper that you basically buckled, except that it actively did this in the domain where twist is being expressed. And so you can also look onto this array of cells on the apical surface, and what you see then is that this looks a lot like, polygonal tiling of the plane. And when you look at this some 5 minutes later, you will actually observe that these cells are now drastically different in their shape. Some of them have actually become very small. And the main player involved in making these shape changes is basically the actomyosin inside the skeleton. And so if you're looking at this cartoon of the cell, where in blue you see the outline of the cell, the cell surface, they're stuck together by adhesion molecules, caderins, and then there are actin filaments that are cross-linked by molecular models walking on them that are going to basically play the main role in generating forces that are changing the size of edges that you can see. And so these motors are basically walking on these filaments in some direction or fashion. If they can cross-link with another motor that is on another filament that is antipolar, then they can basically contract these filaments, and that is how you can then shorten the cell interface. And so this then basically has consequences for the physics that is happening. So we've already seen some experiments in people who demonstrated laser ablation, right? So what we're doing here is we're looking at the cell array where we have 4 cells, and we're going to basically induce an ablation at this interface, and then we're going to measure how those 2 green dots, the vertices of shared by 3 cells, are going to move. And what we almost uniformly see or universally see is that there's going to be a recoil indicative that indeed these junctions are under tension, right? But then we also, so we can conclude that over short time scales this looks like an elastic, right? But then you can also think about sort of this behavior, what happens at a few minutes, right? So what we then have is cells are actually intercalating, right? So they're changing the neighborhood, and so this is kind of the hallmark of some kind of a visco-elastic material, right? And then just for thoughts, right? So in the long run, we are actually not fluid, so we're not just flowing down, but we are staying in our shape, right? And so we need to be solid somehow, right? And so that's something that will come in the future. But what we are thinking about right now is, this regime where we're doing viscoelasticity. Right? And so if you do this, you can basically say that you have a stress-strain relationship where you have some time scale at which the stresses will dissipate, right? And then you can think that this is going to be some kind of a creep flow, right? And you can then basically say, okay, your strain rate is like the velocity that I can measure. If I measure a flow field on the surface of such an embryo, I can then plug these equations together and make some approximations. And what I end up is a hydrodynamic limit of these equations, which is basically going to say that there are 2 terms that are sort of contributing to flow, and 1 term that is the force that is driving this flow.
And these time derivatives are just time derivatives, or they're properly covariant? What are
So
they?
you will have to, in 3D, of course, do everything properly covariant in the long run. And
But right now they are.
right now, this is just a 2D thing that I'm thinking about in flat, right? And then we will look at making these operators covariant, right? And then we have snapshots of the velocity, snapshots of the force field. And what we want to do in the long run is really understand how over time this velocity field goes. And then we have to take all of the work also in the time derivative, right? So properly covariant.
So this is really not only 2D, but it's also flat. Because
This thing is flat. Okay. Flat and no time, right? This is instantaneous, yeah? And we will do it in 3D then in the real way.
And there are a few parameters, you can measure everything?
Unfortunately not, no. So that's the problem, right? So what Maha is referring to is this viscosity, this is an effective viscosity, right? This is an effective bulk modulus, right? And so that's really how we treat this. We treat this as effective parameters that we fit. So I'll walk you through the details in a second. So, okay, so what we can now do in this equation, we had two observables that we would like to get our hand on. It was one the velocity, so the flow field that we see on the embryo surface, and the other one was the force that is driving this thing, right? And we can measure these things maybe, right? So what we have here is basically maps of the flow field on the surface of such an embryo. You know? And you can see basically at very early times, preceding the formation of the ventral furrow, if you make a cut down here in the ventral regions, you will get a relatively simple flow field structure. So you will have a sink on the dorsal pole of this animal, and on the ventral pole you will have a source, and those two pieces are basically associated with one another. The cells are basically moving up towards the dorsal end of this embryo before ventral furrow formation. During ventral furrow formation, we have the same kind of structure, except that the signs have not changed. Yeah? So now cells are moving towards the ventral furrow, and that's actually the time when cells are also moving in. And then afterwards, when we have germband extension, now the flow field has a radically different structure. What you can see here are now vortices and saddles. You know? And so we have to have sort of 4 vortices and 2 saddles for geometric reasons that we had on the shapes of the manifold. Yeah? Okay, and so we would like to be able to explain these patterns now in terms of the distribution of this molecular mode of myosin that I showed you, which is responsible for making the shape changes. Now at the scale of the whole embryo and seeing how far we can actually go, can we understand what's going to happen. And so looking at myosin in this embryo, we can take now cross-sections. These are again the same cross-sections as you've seen on the surface of this other staining, but now we have movies of these cross-sections, right? So this is basically a cut in the middle of the salami, and you can see that this is actually also pretty dynamic. And so what you see in this video, maybe I'll play it in. You can basically see the cellularization front moving in, and there's one myosin pool which is very strongly enriched on the basal part of these embryos, right? So this is basically the basal surface, and there is a lot of myosin on this basal surface, right? And then there's also reported a lot of myosin on the apical surface. It's just not very visible at this point in time because it will come and develop. But here in the ventral region, you can actually see the myosin that is present on the ventral part. Yeah? If you're looking at this basal pool, you can actually see that this is quite strongly asymmetric. So you have low signal in the ventral regions and high signals in the dorsal regions. And so what we can do now with our computational pipeline is we can define surfaces, right? So we can basically define this blue surface and we can define this red surface, and we can map out the distribution of these molecules on each of these surfaces and then see how we can do this. And so this shows you basically a dorsal view on this basal pool of myosin, and this is a lateral view on this basal pool of myosin. And I'm having some insets here so that you can see a little bit how that is like. So basically myosin looks a lot like a sheet down there. You know, it's like a continuous sheet of myosin, except that there are little rings of myosin with holes. And these holes are basically called stalks. They are connecting the cell with the yolk. And we think of this sheet of myosin basically as like a giant contractile rubber, right? And this has a concentration gradient, so you see that on the dorsal pole there's a lot more myosin on the basal population than on the ventral end. And so if you're thinking about this sort of leading to movement, the gradient of this pattern would create the flow, and so that would look somehow similar to the flow field that we've been observing early on, and we make this model something. So then on the apical surface, that was the red surface that I showed you, we also know that there's myosin. Now the myosin there is much more intricate, and I think we have experts here in the house who know a lot about this. So basically myosin is expressed now in these little cables. I don't know if you can see this, the people in the front row can probably see it, where you see very enriched myosin sort of running across multiple cell rows. Huh? And so what we would like to do is basically account for this anisotropic distribution of myosin, and standard segmentation tools that you have at your disposal are not very suitable for this. So what we've done is basically come up with an alternative way of doing this. So instead of segmenting the outline of the cell, which is hard because it's not everywhere labeled, what we're doing is we're using a different approach. We're saying we look at some box inside our tissue, and this is now the zoom onto this box. Yeah? And what you will have in general there are lines that are sort of intersecting, right? And so what we can then do is we can make a radon transform of this pattern of lines into the radon transform space, which then makes peaks appear, and peaks are much easier to find than lines. Yeah? And so this is basically how it works. You have some interrogation line that has some angle and some distance away from some origin, and then you integrate all the signal along this line, which is another good feature because noise is kind of taken out then. And you can move this line around, and you can rotate this line, and it will eventually in this way fall across all the lines that you can have in this space. And what you will then find is basically peaks. And these peaks are going to be proportional to the total intensity that we're having on that line. And from these peaks, we can then measure the orientation that these peaks are in, and then we can basically define a tensorial quantity, which we call the Myosin tensor, that sort of reflects the stresses that are being created by this anisotropic distribution of myosin. Right? And what we can do now is we can basically separate this into an isotropic and a non-isotropic part. Isotropic part would basically be uniform pull in all directions equally, and non-isotropic would be what we're after, and we can measure the magnitude of this. If we're measuring the magnitude of this anisotropic myosin, what we are seeing is that there's actually a relatively strong dorso-ventral modulation. So people were aware of this kind of change along the anterior-posterior axis that was expected. In the head, you have less myosin anisotropy, but what was kind of new is that there's going to be less anisotropic myosin on the dorsal pole than you would have in ventrolateral regions. And that's in fact one of the things that we need in our models in order to drive flow. Okay, so what we can then do is we can sort of write this equation down in more proper terms where we now have a covariant derivative. And we can say, okay, how do we model now basically the forces driving this fluid on this curved surface? And so what we are saying is basically the basal myosin pool is more or less isotropic. And we can take the gradient of this. And since these are images, we have to sort of convert the currency, right? And so this is a fitting parameter, which is a number that never changes. It's just a number. And then the same thing we can do with the apical myosin. The apical myosin is a tensor, so we take the divergence to get a force. And again, there is a fitting parameter here, and it's also just a number that we sort of determine once, and then it's fixed for all times. And then we have basically a force field that we've measured. And we basically have this equation where we have this effective bulk modulus that could change with time. It's the only time-dependent parameter that we are fitting every time. So what we can do then is we can try to invert this equation using finite element method. And what we are then seeing is that we can basically recapitulate many of the hallmarks that we see in the example. So basically this is our embryo flow field that we measured in red. And remember early on there was this sort of sink on the dorsal side. If we're looking at the predicted pattern that we get if we're plugging in the measured myosin pools for the flow, we're seeing that this begins to look very similar. You can account for how similar it is by actually defining a residual that would measure how largely these angles are deviating and how much they're deviating in length, these vector fields. And you can see that it's basically very small, and we can make this quantitative. You will see that you're around about the 10-15% level. You can do the same thing then at later times, for example at 20 minutes. This is what the measurement looks like. So we have these vortices, one in the head region, one in the tail region, then we have these hyperbolic fixed points there. And then we can plug in our myosin distributions that we have measured, and this is what we get out of our inverse method. And what you then basically see is kind of vortices appearing, they're going in the right direction, but what they don't quite do is match up perfectly well in terms of the location. And this mismatch in terms of the location gives rise to an enormous residual that you can see here. Yes?
How does the bulk modulus change? You said you have to...
Enormously, right? So this is an incompressible flow, right? So you have a very, very high bulk modulus, whereas this is a compressible flow, right? And so this is basically very low. And if we wanted to push it really to the extremes, we could make this a Heaviside function, right? Where we just have an enormous amplitude and that would be it. So we would have then two parameters that we only would need to do, right? So that might work. Right. So okay, and we can do this over time, and what you then see is that we're basically, consistently a bit below this 20% residual level, which basically means, given a myosin configuration at any given point in time, we can say something about the movements on the surface that should occur over the next minute. Yeah. Okay. And so we want to now test whether our understanding is actually good, right? And so this is great in Drosophila, because in Drosophila you have many mutants right at your disposal, and you can then take those mutants, plug them into the microscope, and measure them. And so one of these mutants is the twist mutant. And so twist was this gene that was expressed in the ventral regions and is responsible for making the ventral furrow. And this is the lateral ectoderm. Twist is not expressed in the lateral ectoderm. So only in this blue box we have twist, in the green box we don't have it. If we're getting rid of twist, what we observe is that overall the kinetics in these embryos are strongly reduced. So we see that the embryo, once it's gastrulated, goes through this very fast phase of germband extension. That's the wild type. If we're now going into the twist mutants, we see that this is rather slow. And I think Maha and Benedict Sanson have a very nice paper talking about how strain and intercalation rates in the lateral ectoderm of these twist mutants is actually reduced. And that's in agreement with just saying that it's slow, right? And so we wanted to understand where this is coming from and whether with plugging our model we could actually sort of still be good at this, predicting how it goes. And so what you can measure is this basal myosin pool asymmetry, right? Remember that on the dorsal pole there was a lot more myosin. And when you look at this in these twist embryos, this is completely lost. Now it's equal everywhere. So you have basically uniform distribution of basal myosin. And that's what we think is responsible for this strongly reduced flow field early on before ventral furrow formation. But then you can also go and measure the anisotropic pool of the myosin on the apical surfaces. And what you see in the wild type is that this grows with time as ventral furrow goes in and reaches a peak during the onset of germband extension in wild type, right? This is a black curve. You can measure this now in twist, and what you see in twist is that this is actually strongly reduced. And when you're looking at the model fit, to the flow field that we can get, they're comparing to one another. And so what that basically suggests is that these reduced kinetics that we're having on the surface of this twist embryo are due to two effects. So one is there's no asymmetry in the basal pool, which will effectively lead to cells not being pulled anymore to the top, and then there's nothing that flows around. And then the other thing is that there is going to be a reduced myosin anisotropy, which is going to make the intercalation rates slower, basically. Those edges will have a harder time to shorten, and therefore this animal is going to gastrulate with reduced rates. And so what that raises is an interesting question, right? So if you're thinking about this gene twist, which is being expressed in the ventral furrow, right, but not in the lateral ectoderm, and you're now observing that in the lateral ectoderm, actually the distribution of the household gene changes. It's affected by this absence of the ventral furrow. So you would like to understand how that is coming about. And so one of the ideas, follows what Elliot has been talking about, right? It's mechanical feedback. Right? So you could imagine that the ventral furrow kind of pulling cells in strains them a lot. Right? And in order to withstand this strain, you will recruit myosin, right? And this is all going in the right direction because what you have is you have this circle, right? That basically is the cross-section to the embryo. The ventral furrow is going to pull, and those edges along that interfaces are the enriched ones already. So you will just make it stronger by pulling the ventral furrow in when it actually So this is an idea under construction. So to summarize what we've done, so we basically have a bunch of tools that allow us to go to the level of whole embryos, light sheet microscopy, tissue cartography, some automatic data analysis, that together with physical modeling sort of give us an entry point into this question, how we can connect the cytoskeleton to the global organization of an embryo. This goes by some tissue flows, right? And one of the major players that we believe drives this whole process and is extraordinary puzzling is why it's in this thing. It's myosin, and if you know the distribution of myosin, you can predict reasonably well the flow field structure. Of course, this is all driven by genetics. Genetics sets up this pattern of myosin, right? And so eventually genetics will lead to this global organization, and we'll have to do this through configuring this myosin. Right. So as a last thing before I'm coming to an end, I want to make some advertisements. So basically, so I'm coming from Santa Barbara, and I'm wearing pullover because it's never so cold in our place. And so we have a summer school in Santa Barbara, just in case it was hot in summer, that is called Form, Fate, and Function this year, which will be held July 22nd until August 25th, where we try to bring together, much in the style here, different communities to talk to one another, plant community, animal community, and the community of physicists. We will have an experiment, hands-on session in there. So we will have a couple of instructors coming by who will, in a lab, essentially build, whole sort of experiments for you guys, and if students are interested in coming, please apply. We're always looking for people who are interested and excited in sort of working there in years. And then these are the people that I'm grateful working with. And thank you very much for
So you mentioned that you identified a certain amount of apical or basal flows. Should that be interpreted as being sort of mandatory if you know that this is a that in the embryo at the time there are these dynamics of vortices and saddles, or is that something that can be discovered?
Right, so I didn't show you the time evolution of these fields, right? So the basal pool is basically the pool that early on is high and drives the kinetics early on. But this basal pool, as cellularization progresses, diminishes. There's less and less myosin on this cellularization front, and by the time that you have these vortices, the basal pool is essentially complete. There's very little myosin that's in there.
I was wondering whether you had performed laser ablation of the basal myosin to analyze these, whether there is any anisotropy or whether indeed you have this.
Yeah, it's a very interesting idea. It's a very interesting idea.
At the epical side.
Right, so no, we haven't performed this yet, but it's one of the things that is on our menu. One of the problems here is that we are talking about cells that are 35 microns long, and focusing your laser onto something that deep is a bit of a challenge, right? So you need to have high enough energy, well enough focus, so that you can actually ablate, right? And so that's something that we're trying to work on right now. So using adaptive optics in order to think about how we can make this cut and then literally measure what the sort of stresses are.
Last question before
So
lunch.
in the residual, I remember, if I remember correctly, that you're measuring the myosin tensor as a function of time. So is it possible are there ideas for predicting that as a function of feedback to flow?
Absolutely, that's what we're doing right now. So what we're working on is basically the question, how does the myosin tensor evolve as a function of time, right? And so, Farzan is actually involved in this, working on this. But what we are interested in is basically saying there's mostly effects that are going to lead to the configuration of myosin has at a certain point. So there's going to be passive effects like affection, right? So the myosin is in cells and cells are transported. Right? Then there is sourcing, right? So there are gene expression profiles that are sourcing the distribution of myosin. That's another thing. And then you have this mechanical feedback idea, right? That may also do this, which would then couple, like you've been saying, right? Basically couple the flow field back into cytoskeletal organization. And that together then may allow us to predict for a longer time how the myosin field is looking like, and on this machine we basically are from one measurement of myosin through an hour long isolation.
Okay, let's thank all the speakers from the morning. Lunch is upstairs and we'll gather back here.